Okay, so I welcome each and every one of you to our tutorial for today. And we'll be looking at cell division. We'll be looking at cell division. And we'll have a preamble, an introduction, and then we'll look at the cell cycle, and then look at mitosis. So we'll look at the cell cycle and mitosis. Remember, when we started looking at cells, we did mention that the cells are actually the most basic units of life, the basic unit functionally and structurally as well. And so we see that each and every living organism actually is composed of cells. It's actually composed of cells. And we know that cells will build up on tissues. Tissues will give rise to organs, and organs will give rise to systems, and the systems will give rise to an organism. So the cells become the foundational or basic units of living organisms. We did look at the cell theory, in which we also found out that a cell will come out from pre-existing matter or pre-existing other cells. And cells are able to reproduce by copying their genetic information and undergoing cell division. So through cell division, the parent is able to give rise to daughter cells. So through cell division, daughter cells are given out or arise. Okay, so when we look at cell division is very important in that it adds to the population of organisms, especially in relation to the single-celled organisms. Single-celled organisms are able to divide and hence grow in number. When we look at the multicellular organisms, such as human beings, such as you and me, we find that cell division is important for our growth and our development. It is important for replacement and repair of tissues, and also regeneration. So cell division is important for increasing the number of population of organisms. It is important for growth and development. It is important for tissue repair and cell replacement. It is important for tissue and organ regeneration. So for example, the starfish and the salamanders. An example of for tissue and organ regeneration is the starfish and the salamanders. Okay. The other function of cell division, especially in relation to mitosis, is that it leads to the maintenance of the chromosomal number. So it gives rise to genetic stability, And also we have asexual reproduction, such as vegetative propagation that takes place in plants. And we also have budding, which takes place in yeast. So cells can divide for the purpose of growth, for the purpose of repair, and for the purpose of reproduction. So growth and development. So what are the types of cell division? So basically there are three types of cell division. We have binary fission, which we actually looked at when we considered the topic of prokaryotic cells. We did look at a binary fission. And then we have mitosis and meiosis as well. And we are going to consider these, especially the last two, mitosis and meiosis, since we already covered binary fission. So as mentioned earlier, binary fission takes place in prokaryotic cells. But we see that mitosis and meiosis will take place in eukaryotic cells, and we see that these two processes are actually complex and more advanced. So now let's look at binary fusion. Let's look at binary fusion. So we did talk about binary fusion in that it's a special type of cell division which takes place in bacteria and archaea. Remember, we divided prokaryotic cells into bacteria and archaea. And we see that the genetic material in the nucleus is arranged in circular chromosome of the DNA. So in a similar fashion, when we look at eukaryotic, the genetic materials is actually duplicated before cell or before division. The material of these cells is duplicated before division takes place. So as the prokaryote elongates during this process of binary fusion, as it elongates, as you can see in the diagram there, are going to see that the two chromosomes that are attached to the plasma membrane will also move apart. And once these two copies have separated, that is the original chromosome and the replicate chromosome, the cell divides. And this is a process that we call cytokinesis. The process we call cytokinesis. So except in cases of mutation, you find that these two are actually identical. Except in cases of mutation, these two are actually identical. Okay, so let's avoid making sketches on the screen. So this is what is being depicted as binary fusion. Now let's look at cell division in eukaryotes. So as mentioned earlier, we said that the cell division that happens in the eukaryotes is actually more complex and more complicated. It's more advanced, just as the eukaryotic cells themselves are actually more advanced and more complicated. So let's look at the cell cycle, which is being depicted on the two diagrams there. So it's simply a series of events which takes place in a cell, which leads to duplication and or replication. So it's a series of events which takes place in a cell, leading to its replication. So that's a cell cycle. So it's the period from the beginning of one cell division to the beginning of the next cell division. So it's a period from the beginning of one cell division to the beginning of the next cell division. So as depicted there, it's a cycle. So it can be represented as a circle, which is composed of these phases. We have the G1 phase, we have the synthesis phase, we have the G2 phase, and we have the M phase. So we have the G phase, S phase, G2 phase, and the G1 phase, S phase, G2 phase, and the M phase. And this is responsible for planning and for the development of these cells. So we see that the first three stages we see here make up what is known as the interphase stage, where the cell actually spends about 90% of the whole of this cycle. So 90% of the cycle happens in the interphase period, the interphase period. So this is the interphase period. And we are going to dive more into what happens during the interphase period and the cell cycle in general. So the cell cycle is also composed of all the steps that are required for the reproduction of a eukaryotic cell. So let us look at the interphase period. And before we look at that, a concept known as generation time refers to the time that it takes for one complete cell cycle. So for one complete cell cycle to happen, that's what is known as the generation time. The generation time. So that's the generation time. So basically, am I still breaking? Hello, am I
Yes.
audible? Yes. Hello, am I audible? Okay, so when we look at the interphase phase or the interphase period, this is also known as the preparatory phase. It's a preparatory phase and hence takes place before mitosis. So in the cell cycle in eukaryotic, it's basically divided into two periods, and that's the interphase period and the mitotic phase or the M phase. So the preparatory phase is the interphase period. The interphase period. So this takes place before mitosis and cytokinesis. So it's important that before a cell can actually enter cell division, it needs to synthesize different essential molecules. So all those preparations are actually done during the interphase period. And we mentioned that this is composed of the G1, the S phase, and the G2 phase. So during this preparation period, we see that we have those stages. So it's in three stages: the gap 1 phase, which is G1, the synthesis phase, which is the S phase, and the gap 2 phase. So during the process of interphase, there is no cell division. The cell does not divide. So there is no division of the nucleus or the cytoplasm. The G phase, the interphase period simply prepares the cell for division, for cell division. So we are saying in interphase, the cell is prepared for division, for DNA replication. The cell prepares for division via DNA replication, via organelles replication, and via increases in the cell size. So this is basically what happens during the interphase period. This is how the interphase period prepares the cell for division. So through DNA replication, through organelle replication, and through increase in size of the cell. So now we have to look at the stages or the phases. So I said that we have the G1 phase or the gap 1 phase. So now what happens during the G1 phase is that during this phase, which is the first phase of interphase, and also the first growth phase as well. So what happens during the G1 phase is simply synthesis of enzymes and other proteins. So this phase prepares for the synthesis stage by making sure that all materials required for DNA synthesis or replication are present. So this means that essential proteins, enzymes are synthesized during this period of the G1. So there's gathering of nutrients and it's a gap between mitosis and DNA replication. So you can see the G1 phase as shown on the diagram there. That's a G1 phase, the G1 phase. And you can see this is DNA replication and this is mitosis. So it's occurring between mitosis and the DNA replication. So there's gathering of nutrients during the G1 phase. So I've already mentioned that the synthesis of RNA and proteins that are necessary for DNA synthesis and replication. So that's a G1 phase. Now, what happened during the S phase? So the S phase is also known as synthesis phase. Synthesis phase. So during this period, we see DNA and chromosomal replication taking place. DNA and chromosomal replication taking place. So DNA is copied so that the daughter cells will have a complete set of chromosomes at the end of the cycle. So this is the period of DNA synthesis. No wonder it's called the synthesis stage or the S phase. So there's a replication of DNA as mentioned earlier. So at the beginning of this phase, the chromosome number is 2N or diploid, and the DNA content is also 2D as well. At the end of this phase, we are going to say that the chromosome number will remain constant. The chromosome number will remain constant while the DNA content will actually double. DNA content will actually double.
So could you go back to the previous slide?
And then we have the G2 phase. We have the G2 phase or the GAP2 phase. Please, shall we mute ourselves?
Sure.
So we have the G2 phase. Hello.
We are again asking if you can bring the first one, the gap one.
Yes, what question do you have on that?
I didn't get clearly on the DNA part.
On the DNA part. So under gap one, what is important is that this is the first part, the first phase of interphase, and it's the first growth phase. Secondly, during this gap one, this is when the cell is mainly synthesizing enzymes and other proteins that are essential for DNA replication. That's the G1 phase. And then during the S phase, this is where there's DNA and chromosomal replication taking place. And we mentioned that at the end of this S phase, what we are going to have is that each chromosome will have 2 sister chromatids, while the DNA content will actually double. So that's what you need to take note on the G1 and S phase. Furthermore, on the S phase, you find that this process is actually has to happen quickly so that there's avoidance of damage to the DNA. And therefore, to minimize any vulnerability of DNA mutations taking place. So those are what are essential on the G1 and G2, the G1 and the S phase. And then after that, we have the G2 phase, which is simply the second growth phase, the second growth phase. So during this phase, the cell is preparing for mitosis and meiosis. So it makes sure that all the raw materials that are required for the physical separation are present. all the materials that are required for the physical separation are present. That's what happens during the G2 phase. It's a second growth phase. Remember, the first growth phase is G1. So G2, it's a second growth phase. So it's the gap between DNA replication, as we can see, DNA replication and mitosis. DNA replication and mitosis. So there's growth of the cell during the G2 phase. So organelles replicate, and chromosomes also coil and shorten as well in readiness to enter the mitotic phase or the M phase. So that's what happened during the G2 phase. And then we have what is known as the quiescent stage or the G0 stage.